All right. Hey, everyone. My name is Yajat. One of the new members of TS Platypus this year. Super excited to be part of the team. Feel free to message me with any questions you may have throughout the year about any content or anything. It's all good. Today, we'll be talking about lung function tests. Let's get right into it. So we'll start off with dyspnea, which is a fancy word for shortness of breath. And if someone comes in with shortness of breath, you're thinking of a bunch of different causes for it. First example would be deconditioning, which is just a bunch of physiological changes that happen after being inactive for a while. Cardiac causes, your heart failure, your AMI, they can all present with shortness of breath. There's not enough blood being pumped around the body. So people get hypoxic and have trouble breathing. Anemia, your heart's pumping properly, but there's not enough red blood cells carrying the oxygen around. Again, you can very traditionally get shortness of breath. But what we'll be focusing on today and for the next few weeks, are pulmonary or respiratory causes. And there's a whole bunch that can present with shortness of breath. And so they're mainly split into obstructive conditions or restrictive conditions. In obstructive conditions, patients have trouble getting air out of the lungs, and you get this thing called air trapping. So as you can imagine, the function of lungs is bringing air into the body and bringing oxygen in and carbon dioxide out. And so if you can't get air out of the lungs, you can imagine there'll be poor oxygenation. Restrictive, on the other hand, is the complete opposite. You can't get air into your lungs, and fairly intuitive, poor oxygenation once again. So this is an example of air trapping that I was talking about in an obstructive picture. You've got a chest X-ray here. Major landmarks have got your heart, your vertebral column, clavicle, scapulae, diaphragm at the bottom, and all this hypodense region is your lungs because it's filled with air. So normally in a normal picture, what you want to see at the bottom of your lungs are these nice curved diaphragms. Your right one higher than your left because of the liver under the right side. But what we see that in this pathological state is flattening of the diaphragm. So there's not as much of a curve to them. And this happens in a condition known as COPD, chronic obstructive pulmonary disorder, which is as in the name, it's an obstructive condition. And so you get air trapping and hyperinflation of the lungs, and they press down on the diaphragms and flatten them. So it's a very traditional, very classic sign you can see on the x-ray, so keep an eye out for it. If you see this, you're thinking COPD or obstructive disorders, flattening of the diaphragms, yeah. So how do we distinguish between obstructive and restrictive disorders? They both present with shortness of breath. Is this machine over here called, or this investigation called spirometry. So you have the patient breathe into this tube that's connected to a computer, and it's measuring how much air is going in and out. And essentially you ask the patient to first breathe normally, ask them to take a big deep breath in as much as they can, and then breathe out as hard as they can and as much as they can. And you need them to breathe out for almost 6 seconds for it to be a viable result. And after the 6 seconds, you get these two values. You get FEV1, which is the amount of air that's blown out in the first second of forced expiration. So if you give it a shot, you'll notice that most of the air that you blow out will go out in the first second, and there's not much left after that. And FVC is the total volume of air over the 6 seconds or however long the patient takes to blow all the air out. And so that's a total volume of air on forced expiration. And what's important is the ratio between these two values. So your FEV1 over FVC ratio, normally around 70 to 80%. So normally around 80% of your air is blown out in the first second. And changes in this ratio can be used to diagnose obstructive versus restrictive diseases. So here's a look at a normal spirometry graph that you'd get after you go through it. And you can see the steep incline in the first second. You're getting almost all the air out. You go volume on the y-axis, time on the x-axis. In the first second, you get almost 80% of your air out, and then it tapers off, and the total amount of volume is your FVC. Here are your normal ranges. Your FVC normally is around 5 liters, the amount of air you can get out in forced expiration, and the 4 liters of that is expired in the first second, giving you a ratio of around 0.8. It's the normal value, magic number you're looking for. So here's what happens in pathological states. So you got normal in blue over here, just like very similar to what we saw before. And in restrictive, you can see two things. First of all, your plateau or your FVC is significantly lower than your normal. But what's more important is that it's still following the same path. Because in restrictive, if you remember, there's no trouble getting air out of your lungs. all the issue is getting air into your lungs. So there's less air coming into your lungs, but in expiration, that still follows a normal rate. And so your FEV1 over FVC ratio should be normal since it's following the same curve. Compare that to an obstructive condition where all the issue is in getting air out of your lungs, and you'll see this very gradual incline. And so in your first second, you've barely gotten any of the air out because that's where the issue is. And so you notice a big drop in the ratio there. So yeah, for a visual representation, your FEV1 drops in both these conditions, but it drops far more in obstructive conditions. Compare that to FVC, which also drop in both the conditions because your lungs just aren't functioning right. But there's not much of a difference between restrictive and obstructive there. So yeah, the ratios. This is a summary table. Your obstructive, the ratio decreases. In restrictive, it's normal, or it may even increase. And you can go through these values, but they're fairly intuitive. Your FEV1 is decreased substantially in obstructive. There shouldn't be too much of an issue of your FVC. But what's important in this table is at the bottom are the examples of the actual conditions or pathologies that can present as obstructive pictures or restrictive pictures. So obstructive is fairly easy to remember. Your asthma and your COPD, you get the lecture together. They're pretty similar in the way they present in the sense of spirometry. Whereas your restrictive, there's a bunch of different conditions that can cause it, and they vary greatly. I mean, you've got spinal deformities, which is a very mechanical issue, but your lungs just can't inflate and deflate regularly. Compare that to fibrosis or ILD, interstitial lung disease, where that's more of an at an alveolar level. There's issues with diffusion across the membrane. And so we'll go through how to differentiate between all these different conditions in further investigations in a minute. All right. So these are a bunch of different values that you should know, and we'll go through them right now. And it might seem overwhelming, but I think if you just understand this diagram, it should be fairly intuitive. So we go through a patient who presents with this diagram on the right-hand side. You ask the patient, again, breathing into spirometry, into the tube, you ask them to first breathe normally. And they inhale, they exhale, they inhale, they exhale. It's not fluctuating too much. You don't need to inspire or expire too much air for normal lung function. So this is known as your tidal volume, the amount of normal, quiet breathing breath. You then ask the patient to forcibly breathe out as much air as possible. And you see this big dip. And this is your maximum possible expiration. And this is known as your expiratory reserve volume. Then ask them to breathe normally back to tidal volume. And then you ask them to do the opposite. You ask them to take a big, deep breath in. And that's your inspiratory reserve volume. You see the volume increases rapidly and then comes back to normal. So these are fairly intuitive. Tidal volumes, normal, quiet breathing, inspiratory reserve volume, expiratory reserve volume. Now, one thing to notice is at the end of maximum possible expiration, which is voluntary expiration, there's still a bunch of air left in the lungs. You still need air in your alveoli to keep the airways patent, otherwise your lungs would just collapse. And so you've got this around 1.2 liters of air still in your lungs, and that's known as your residual volume. So the amount of air left after forced expiration. If we then take everything we've talked about now, so from top to bottom, that's known as your TLC, or total lung capacity, just the amount of air in the lungs. If we then take from the bottom of maximum possible expiration, so the voluntary change is in the amount of air to maximum possible inspiration, that's known as your vital capacity. So your total lung capacity minus your residual volume. There's different ways to look at it, different formulas you can look at. And that's pretty much it. Inspiratory capacity is not that important. FRC is your functional reserve capacity. And that's the amount of air left after expiration of tidal volume. Just something I guess you need to know. But if you go through this diagram and you understand these values, it should be fairly intuitive. Now, if we look at what spirometry can measure, now I talked about this being a relatively theoretical value since we can't really measure it, the residual volume. And so anything below the maximum possible expiration, you can't really measure in spirometry because that's the amount of air left in your lungs. So you can measure anything above that, your vital capacity, your inspiratory reserve volume, your expiratory reserve volume, your tidal volume, all this stuff is able to be measured just through that tube on the computer. You can't measure anything below that. Your residual volume, your functional reserve capacity, your total lung capacity. And you really actually need to measure them. They don't have much applications clinically. There are some special techniques that do exist, not that important. Yeah, these are just some formulas that are worth knowing. But if you do understand this and the relationships between all these different capacities and volumes, it should be fairly intuitive. All right, DLCO, it's another lung function test. This time we're looking at alveolar function. So what's a function of alveoli for diffusion of oxygen and carbon dioxide across the membrane? And so the way it's done is sort of complicated. I don't think you really need to know it much. You sort of are just told if it's decreased or if it's normal. But essentially, you've got some carbon monoxide, which measures diffusion. You've got some helium, which measures volume. Breathe it in, you breathe it out. The concentration of the gases are measured before and after. And that apparently tells you how well your alveoli are working. And so if we go back to restrictive conditions, I was talking about earlier, so you got your fibrosis, your interstitial lung disease, your pulmonary edema. Those are issues at the alveolar level. And so you can imagine that diffusion across the membrane would be impaired. And so you'll have a decreased DLCO there. Whereas something like spinal deformity or chest wall deformity, that's a very mechanical thing. And your lungs just aren't inflating or deflating as they should. And so your alveoli should be functioning normally. And so your DLCO, even though it's a restrictive picture on spirometry, your DLCO should still be normal. And that's how you can differentiate between those. Flow volume loops is, I think, the last investigation you need to know. You've got this diagram here. We'll go through it. We don't have time on the x-axis anymore, which is a bit weird, but time sort of goes in this loop over here. But you're essentially looking at flow, so the speed at which air is going in and out of the lungs. So inhalation is here on the negative y-axis. As the patient inhales, they go over here, the flow increases and it decreases. And then eventually they breathe out. And again, it's a similar shape. And at the top over here, the fastest that the air is being exhaled is your peak expiratory flow. But what you mainly need to know in this is the pathological conditions and how they present on flow volume loops. So your obstructive and your restrictive disorders. So over here, the big loop on the outside is your normal. In obstructive, you have this thing called coving. So if you look at on the exhalation side, you have this big dip as opposed to your normal where it's more gradual decline in flow. You have this big decrease in the speed at which the patient is exhaling air. And this coving is a major sign that you look out for in an obstructive condition, which is caused by reduced airway diameter. Restrictive has a similar shape to your normal, but it's just smaller and skinnier. All right, and this is just a high yield summary. I think PS Pingu made this last year. Very useful flow chart. And if you just go through it, so essentially you start off by looking at the ratio, FEV1, FEC, as we discussed. If it's decreased, you're looking at obstructive. If it's normal, you're looking at restrictive conditions. And as you go down, you'll then, what's important is over here. So this DLCO. So when you've found out that it's a restrictive picture, you want to look at DLCO, and that helps differentiate your ILD, your fibrosis, to where your DLCO decreases to your more mechanical chest wall disorder, obesity conditions where your DLCO should be normal. All right, I think we have a few questions. So yeah, I guess just pause, give it a shot, and we'll go through it. So we have 3 male subjects, same body size, same age, and you get these values in spirometry, these 3 values, and yeah, FEV1, FEC, just the definitions there. So FEV1 in the first patient is 4 liters and FEC is 5 liters. If you remember, that's our normal picture. Those are normal values. You've got a ratio of around 0.8. So patient 1 is normal lung. So you're already down to A, B, or D. If we look at patient 2, you've got an FEV1 of 1.3, FEC of 3.3, which is approximately 1/3. So that ratio is dipped quite a lot. And so then you're thinking, when does the ratio decrease? It's an obstructive lung disease. So patient 2, obstructive lung disease. And patient 3, FEV1 2.5 liters, FEC 3 liters. That's around 5/6, which 1/6 versus 1/5. So the ratio is increased. It's more than 80%. And so if you remember, if restrictive lung disease, the ratio is either the same or increased. So normal, obstructive, restrictive, we're thinking D as the answer. Sweet. A direct reading from a spirometer can only be used to measure total lung volume. That's from the top to the bottom. So you can't remember if you can't measure the amount of air left in the lungs after forced expiration. So that's a no. Residual volume, the amount of air left in after forced expiration, still a no. Functional residual capacity, that's from the bottom of tidal volume all the way down to 0. So we still can't measure that. Total lung capacity, pretty much the same thing as total lung volume. And that leaves inspiratory reserve volume, which is the amount of air, the volume of air breathed in with forced inspiration with maximum inspiration. So we can measure that. It's the basis of a lot of spirometry. So we'll go with either. Nice. This is a restrictive or obstructive picture. We're given TLC, RV, FEV1, FEC, and the ratio. All you need is the FEV1 over FEC ratio. It's normal, maybe slightly increased. That is restrictive. So that's really all you need. And all these values sort of make sense in a restrictive picture. Yeah, so. I think second last question, is this restrictive or obstructive? There are flow volume loops. What are we looking at here is the coving on the expiration side of the flow volume loops. Since there you see this big dip, you're thinking obstruction, having trouble getting air out of the lungs. There, obstructive, big dip in the positive y-axis. And last question, what do these spirometry results indicate? You've given normal values, and this can be a bit hard to visualize. You're not presented in the same way you're expecting, but faculty does present questions like this sometimes, so it's good to get used to. FEV1, 2.5 liters, FEC, 3 liters. Around 5/6 as the ratio, which is increased, as we said before, so we're thinking a restrictive picture. Whoops. Sorry, my screen just locked. Asthma is not a restrictive condition, so we can get that. We can get rid of that. Mixed is, I don't even know how to differentiate mixed, so it's not that important. And so you have your two restrictive conditions, interstitial lung disease and chest wall disorder. How are we going to differentiate between these? Remember, interstitial lung disease is disease around the level of the alveoli. Your alveolar function will be decreased, so you're expecting a low DLCO. Whereas chest wall disorder is a mechanical function. Your lungs can't inflate and deflate properly, but your alveolar function should still be normal. So DLCO should be normal. Here we're given DLCO is low, so we'll go with ILD A. All right, that's about all I've got for you today. I'll see you next week then.